In this video, we are going to talk about DNA replication and the experiment of Winesett and Stahl. When their experiment was conducted in 1958, 3 hypotheses for DNA replication had been proposed: conservative, dispersive, and semi-conservative. In the conservative model, a new copy of DNA is made in the first replication. In the dispersive model, the first replication results in 2 DNA molecules that have a combination of original and new DNA. In semi-conservative, the new DNA has 1 original strand and 1 new strand. To determine which hypothesis was correct for DNA, Winesett and Stahl grew a culture of E. coli cells in 15N media. Then, they transferred the cells to a 14N media and let the cells replicate for several generations. The main difference between 15N media and 14N media is in their weight, and 15N is heavier than 14N. After each generation replicated, they took a sample of the DNA. The DNA samples were centrifuged, which means that they were spun so the different DNA strands could be separated by their weight. They observed that the DNA in the 15N media produced 1 15N DNA band. After the first replication in 14N media, 1 band was produced that was a combination of 15N and 14N, as seen here in purple. After the second and third replications in 14N media, 2 bands were produced. 1 band was a combination of 15N and 14N, as seen again in purple, and the second band was 14N, as seen in red. Referring back to this chart, Winesett and Stahl could rule out the conservative model after the first replication because a sample of DNA did not produce 1 15 band and 1 14 band. However, a second generation of replication was needed to distinguish between dispersive and semi-conservative because after the first replication, they both produce a purple band, which is a combination of 15N and 14N. After the second replication, they ruled out the dispersive model because 2 bands were produced. 1 band was a combination of 14N and 15N, and the second band was a 14N. If replication was dispersive, then all that would have been produced was just 1 14N and 1 15N band, which would have been in purple. Therefore, they were able to identify the replication of DNA as semi-conservative. In conclusion, DNA replication is semi-conservative, which means that each strand of the original double-stranded DNA is used as a template for replication so that every piece of DNA will have 1 old strand and 1 new strand.